Have you ever wondered how our cells duplicate their DNA? It's a fascinating process that lies at the heart of life as we know it. DNA, or deoxyribonucleic acid, is like the blueprint of life. It carries the instructions our bodies need to develop, grow, and function. Every cell in our bodies contains this vital DNA. But each time a cell divides, it needs to make a copy of its DNA. This is where DNA replication comes in. It's a careful, intricate process that ensures each new cell has the same set of instructions as the original. This happens during a phase of the cell cycle known as the S, or synthesis phase. DNA replication isn't just about making copies though. It's also a key player in maintaining the integrity of our genetic material, helping to keep us healthy and thriving. Understanding how our cells duplicate their DNA is key to understanding life at its most fundamental level. Now that we have a basic understanding of what DNA is and why it needs to replicate, you might be wondering, how does this process actually occur? Let's delve into it. DNA replication is a complex process that begins when an enzyme called helicase comes into play. Picture helicase as a tiny molecular motor. It acts on the DNA molecule, breaking the hydrogen bonds between the base pairs that hold the two strands of the DNA double helix together. This action of helicase unwinds the DNA, separating it into two single strands. As you can imagine, this creates a Y-shaped structure known as the replication fork, which is the active area where DNA replication occurs. Now, on to the next step. Here's where RNA primase steps in. RNA primase is an enzyme that synthesizes a short RNA segment called a primer as a starting point for DNA replication. Think of this primer as a launch pad for the next big step in our replication process. Once the primer is set, the enzyme DNA polymerase comes into action. DNA polymerase is a crucial player in this process. It's like a builder, adding nucleotides, the building blocks of DNA, to the growing DNA strand. DNA polymerase does this by reading the sequence of the original DNA strand and adding complementary nucleotides to the new strand. This is done in a very particular manner, always adding nucleotides to the 3' end of the new growing strand. But here's something interesting. DNA polymerase can only add nucleotides in one direction, from the 5' end to the 3' end of the new strand. This means that one of the strands, named the leading strand, is synthesized continuously, while the other strand, known as the lagging strand, is synthesized in fragments. So by the end of this part of the process, we have two strands of DNA, each with a primer and a section of new DNA. But guess what? That's just the start. That's the first part of DNA replication, but it's not over yet. We've come far in understanding the process, but there's more to DNA replication. Just as we left off, the lagging strand is being synthesized in fragments, known as Okazaki fragments. These fragments are not immediately connected. This is where DNA ligase comes into play. Acting like a molecular glue, DNA ligase joins these fragments together, sealing the nicks between them and creating a continuous DNA strand. Now, let's look at the bigger picture. The DNA molecule has been unzipped and both strands have been copied. But what's unique about DNA replication is the result. At the end of this process, there are two identical DNA molecules. Each of these molecules contains one strand from the original DNA and one newly synthesized strand. This is referred to as semi-conservative replication. Why semi-conservative, you ask? Well, because each of the two resulting DNA molecules conserves one of the original strands and includes one new one. It's like getting a copy of a book where half the pages are from the original book and the other half are freshly printed. This process ensures that the genetic information is accurately passed on to the next generation of cells. DNA replication, in all its complexity, is a fine-tuned process that allows life as we know it to continue. And just like that, our cells have duplicated their DNA. So, to recap, let's quickly summarize the process of DNA replication. It's a two-step dance, really. First, enzymes unzip the DNA double helix, creating a replication fork. Then, DNA polymerases swoop in, using the original strands as templates to synthesize new ones. This process is repeated until the entire DNA molecule is replicated. And that, in a nutshell, is how our cells duplicate their DNA. Keep wondering, keep learning, and remember, every cell in your body is a testament to this amazing process.